Let's talk about transcription. Now you might wonder why I'm bringing this up when I've been talking about metabolism, but it does actually relate, and it covers some of the questions such as, why do muscle cells and liver cells store glycogen and fatty acids, but nothing else in the body seems to? Well, what's important to remember is that the way that fatty acids and glycogen are stored is by using enzymes that synthesize them, okay? And the reason they get stored is because they're a relatively low energy, well, I say low energy, they they contain a lot of energy in their bonds, but they are not easily broken down. They require specific enzymes that have been activated in order to break them down. So that would be beta oxidation for fatty acids, and that would be glycogenolysis for breaking down glycogen. So why do muscle cells store glycogen? Why do liver cells store glycogen? And why does the brain not store glycogen? It's because the enzymes that are necessary for storing glycogen, for synthesizing glycogen, and for breaking it down are present in muscle cells and liver cells, not in the brain, not in the skin, etc. Okay, so the way that this sort of transcriptional regulation works is that you can see here my crude drawing of a cell, okay, with a nucleus. I just want to go over, but I hope that we're all comfortable with the fact that these membranes, or this membrane, sorry, is a lipid bilayer. This is another lipid bilayer. Okay, that means that you have this hydrophilic head and then hydrophobic tail, and you have a bunch of these lined up, right? And then they make this bilayer, and this is a sort of semi-permeable membrane where only certain molecules can pass through. Okay, so that's what those two membranes are made up of. That's important when it comes to cell signaling because only small molecules can get through, but a lot of larger proteins like albumin, for example, and bilirubin, things like that, cannot get through this cell membrane without some sort of transporter. That will also be important later when we're talking about glucose uptake from the blood, for example, and why the brain can only eat glucose, things like that, why muscle cells can work off of other things like fatty acids. Alright, so we have our cell, and what you should all remember, hopefully from genetics, is that every somatic cell in our bodies have the exact same makeup, right? They contain the exact same chromosomes, the exact same DNA. What makes a muscle cell different from a liver cell, different from a skin or a brain cell, is the factors that are transcribed from that DNA. So I hope that we're all comfortable with the fact that not 100% of our DNA is transcribed. When I say transcribed, I mean converted into mRNA in order to be translated into protein. Okay, so we've got our representative piece of DNA. Okay, this white space is a gene, and this shaded space is the promoter for that gene. I don't know how much of this was covered in detail in anyone's biology background, so I'm going to go through it anyway. Genes have promoters in front of them. They have regions of DNA that are not converted into protein, but are important in order for the transcriptional control of that gene. That is, there are protein factors that can bind to this non-coding region of DNA in front of the gene in order to turn transcription up or down, on or off, okay? In the same way that we were talking about enzymes being able to be turned on or off, up or down, gene transcription is regulated in a similar way, okay? It can be turned up or it can be turned down, or it can be shut off entirely. So the way that this sort of generally works is if we're talking about a muscle cell wanting to produce more glycogen synthase in order to generate more glycogen, what you're going to have is some sort of receptor on the cell surface. Okay, so it's got this binding domain out here, and then it will generally pass several times through the membrane. It will be embedded in the membrane. Okay, embedded meaning that it has a cytoplasmic domain on the inside. That's what this tail is right here. I'll put a bigger piece on it so that it's a little clearer. This is the cytoplasmic domain. Okay, and this is the extracellular domain. That means that it's the piece of the protein that exists outside of the cell, but it is still bound within this membrane, okay? So you're going to have some sort of molecule in this extracellular matrix. Maybe it was secreted by another cell, maybe it's a hormone that is being taken up from the bloodstream, and it's going to bind to this receptor. It has a particular shape, it will fit a particular receptor. Not all cells will have these receptors, and so not all cells will bind to this molecule. Okay, the molecule, the ligand, let me write that down. Ligand is specific for this receptor. Okay? So once the ligand binds, okay, some sort of conformational change will take place within this protein, because what you need to remember is that proteins are really just amino acids stuck to each other, and amino acids are just atoms that are chemically bonded. And when we say chemically bonded, they're bonded by electrons, which means that there are electrostatic interactions, that is, positive and negative charged reactions, or interactions, going on within this larger protein. Okay, there's charged regions, there's uncharged regions, and when you have this binding action up here, that changes the electrostatic interactions going on within the protein, and therefore changes the conformation of that protein. So instead of having that tail pointing that way, maybe you have a tail pointing this way. Okay, and the shape of it changes. That is going to cause this signaling cascade to happen. There will be a number of factors, proteins usually, affected by the change in conformation of this one part of this one protein, in this case a receptor. So there's this cascade, it's called a signaling cascade, of protein changes, modifications, and you might eventually have a transcription factor, okay, TF for short, transcription factor. That's simply another protein. Say that this ligand eventually, through the signaling cascade, causes the synthesis of this transcription factor, or increases the availability somehow of this transcription factor. Well, that factor, that protein, is transported into the nucleus, and it can bind specifically to this promoter, okay, this promoter region. Again, this binding is specific. There are specific contacts, locations, where this transcription factor will bind, and it will bind to one promoter but not another promoter. So now that you have binding, that activates the transcription of this stretch of DNA, this stretch of coding DNA, right? And then you get mRNA, right? This part should be familiar to everyone. The mRNA gets translocated to the cytoplasm and is translated into protein, okay, by the ribosome. And when I say it's translated into protein, what we're really talking about, or the specific example that I was using, was with glycogen synthase. So now that the signaling cascade has happened, this transcription factor has been recruited, it binds to the promoter and produces more glycogen synthase. Now that you have a higher availability of this enzyme, the process of glycogen synthesis in this particular muscle cell will happen faster, okay, because there's more opportunity for binding between enzyme and substrate. That is one mechanism by which reaction speed, pathway direction, etc. can be controlled.